Morning students, we are discussing steps involved in genetic engineering process. Right? In this we completed number 1, isolation of desired gene. Number 2, insertion of desired gene into vector. Number 3, introduction of rDNA into the host cell. Number 4, there is a selection of transformed cells. We are discussing selection of transformed cells. In selection of transformed cells, we completed by using one antibiotic resistant gene. We completed by using 2 antibiotic resistant genes. Now we are discussing insertional inactivation. What's that? Insertional inactivation. Sir, this insertional inactivation also called as a right, blue white selection. Also called as what, sir? Blue white selection. Is it okay? Right? Now we are discussing what, sir? Insertional inactivation. Okay, right? Now listen here. Sir, take a bacterial cell. For example, sir, this is a bacterial cell, sir. This is which cell, sir? Bacterial cell. This is a bacterial cell, right? In this bacterial cell, right, for example, this is a plasmid. What is this one? This is a plasmid. Plasmid means double-stranded DNA, right? Plasmid means what, sir? Double-stranded DNA. Plasmid means what? Double-stranded DNA. Sir, in DNA, segments are called as what, sir? Genes, right? For example, this is one gene. Okay, right. What is this one, sir? This is a gene. Sir, when this gene is expressed, a protein is produced, sir. When gene expressed, what is produced, sir? Protein is produced. What is produced? Protein is produced. Sir, this protein, it acts as an enzyme. This protein, what, sir? Acts as an enzyme. What is that enzyme name, sir? Acts as an what, sir? Enzyme. Acts as an enzyme. What is that enzyme name, sir? Beta galactosidase. Beta galactosidase. In shortcut, we are writing this enzyme as beta gal. Is this statement okay? Right. Sir, this is a bacterial cell. In bacterial cell, this is the plasmid. In plasmid, this is one of the gene. When this gene is expressed, a protein is produced. This protein acts as an enzyme by name beta galactosidase. What is the enzyme name? Beta galactosidase. Sir, what this enzyme do, sir? Which enzyme? Beta galactosidase enzyme. Sir, what this beta galactosidase enzyme does means it converts chromogenic substrate into product. It converts what, sir? It converts chromogenic substrate. Converts what? Chromogenic. Converts chromogenic substrate. Chromogenic what, sir? Substrate. Converts chromogenic substrate into what, sir? Blue colored product. Converts chromogenic substrate into what, sir? Blue colored product. Is this statement okay? Right? Sir, who is converting, right, chromogenic substrate into blue colored product, sir? Beta gal enzyme. Here statement is important. Sir, to get a blue colored product, what are the requirements? Number 1, beta galactosidase enzyme should be there. Number 2, chromogenic substrate should be there. Sir, chromogenic substrate is there, sir. but beta gal enzyme is not there, sir, you won't get blue colored product. Sir, beta galactosidase enzyme is there, sir. Chromogenic substrate is not there, sir. You won't get blue colored product. Therefore, to produce what, sir? Blue colored product. To produce what, sir? Blue colored product. To produce blue colored product, right? Beta galactosidase enzyme should be there. Chromogenic substrate should be there. Sir, what is the meaning of chromogenic substrate? Name itself indicates chromo means color, gen means produce. Chromo means color, gen means produce. It means when the substrate is converted into product, we are getting the color. Is it okay? Right. Best example for chromogenic substrate. Best example for what, sir? Chromogenic substrate is a 5-bromo. 5 what? 5-bromo 4-chloro beta galacto pyrinoside. Is it okay? 5-bromo 4-chloro beta galacto pyrinoside. Right. This is an example for chromogenic substrate. This is an example for chromogenic substrate. Now listen to the statements carefully. Sir, this gene produces which enzyme? Beta galactosidase enzyme. When this enzyme is present, chromogenic substrate is converted into blue colored product. Yes or no? Now listen to the statement. Sir, this is the gene which produces beta galactosidase enzyme, right? Right. Sir, the gene which produces beta galactosidase enzyme, in the place of that gene, the desired gene is inserted. What we are inserting? Desired gene. Where we are inserting the desired gene? We are inserting the desired gene in the place of the gene which produces beta galactosidase enzyme. Now you tell me, here what is present? Here desired gene is present. Instead of what? Gene which produces beta galactosidase enzyme. Is this statement okay? Sir, if your desired gene is present, so automatically it becomes what, sir? rDNA. Becomes what, sir? rDNA. Then we can call this cell as what, sir? Recombinant cell. This becomes what, sir? Recombinant cell. Becomes a recombinant transformed cell. Becomes which cell, sir? Recombinant transformed cell. Yes or no? Now you tell me. Sir, here what is inserted? Desired gene is inserted. If desired gene is inserted, then the gene which produces beta galactosidase enzyme, that gene is inactivated. Because the desired gene is inserted in the place of the gene which produces beta galactosidase enzyme. It means now beta galactosidase enzyme producing gene is not there. Desired gene is present there. Therefore, when desired gene is inserted in the place of gene which produces beta galactosidase enzyme, then the gene which produces beta galactosidase enzyme gets inactivated. So, enzyme is not produced. As enzyme is not produced, the chromogenic substrate is not converted into blue colored product. It means if blue colored product is not formed, enzyme is not produced. Enzyme is not produced means the gene which produces this enzyme has inactivated. Why it has inactivated? Due to the presence of desired gene. If desired gene is present, the cell becomes what, sir? Recombinant transformed cell. Therefore, no blue color becomes a recombinant transformed cell. For example, sir, desired gene is not there, sir. It means beta galactosidase enzyme producing gene is there, sir. Then automatically this gene expressed produces beta galactosidase enzyme, converts chromogenic substrate into blue colored product. So automatically, right, if blue colored product is formed, enzyme is present. Enzyme is present means this enzyme producing gene is present. It is not inactivated. Not inactivated means their desired gene is not there. Desired gene is not there means is it recombinant or non-recombinant? It is non-recombinant. Is this statement okay? Right? Clear, right? This is what, sir? Insertional inactivation, right? Also called as blue-white selection. Also called as what, sir? Blue-white selection. Sir, here what is happening, sir? Right? When desired gene, what, sir? When desired gene is inserted where? In the place of the gene, in the place of the gene. Which gene? In the place of the gene which produces beta galactosidase enzyme. See this statement? What we inserted? We inserted the desired gene. Where we inserted this desired gene? In the place of the gene which produces beta galactosidase enzyme. Is it okay? Then what happens? Then what happens, sir? Enzyme produced, not produced, not produced. Therefore, beta galactosidase enzyme producing gene, active or inactive? Inactivated. So what happens? Beta gal enzyme produced, not produced, beta galactosidase enzyme not produced. As enzyme is not produced, right, chromogenic substrate not converted, be careful, not converted into what, sir? Blue colored product, not converted into blue colored product. If blue colored product is not obtained, what it indicates, sir? What it indicates? Recombinant transformed cell. Is this statement clear? Okay, right? Therefore, sir, we inserted a desired gene, sir, where in the place of the gene, which produces beta galactosidase enzyme. When we inserted the desired gene in the place of the gene which produces beta galactosidase enzyme, then enzyme producing gene, beta galactosidase enzyme producing gene is inactivated. So we are naming this process insertional inactivation. What is inserted? Desired gene. What is inactivated? The gene which produces beta galactosidase enzyme was inactivated. Sir, as this enzyme became inactivated, as the gene which produces this enzyme becomes inactivated, beta galactosidase enzyme is not produced. As enzyme is not produced, even though chromogenic substrate is present, it is not converted into blue colored product. As blue colored product is not formed, we are saying that it is what's a recombinant transformed cell. Or we are calling it as recombinant transformed cell. Is this statement okay? Right. Sir, if desired gene is not inserted, if desired gene not inserted, desired gene is not inserted, where desired gene is not inserted in the place of gene which produces beta galactosidase enzyme. Now you tell me, sir, here gene is inserted, not inserted, not inserted. If gene is not inserted in the place of the gene which produces beta galactosidase enzyme, then gene which produces the enzyme, which produces beta galactosidase enzyme, active or inactive? Active. As this gene is active, right? It produces what, sir? Beta galactosidase enzyme. What is produced? Beta galactosidase enzyme is produced. What this beta galactosidase enzyme will do, sir? It converts. What it converts? It converts a chromogenic substrate. Converts what, sir? Chromogenic substrate into blue colored product. Therefore, now you tell me, if blue colored product is formed, enzyme is produced. Enzyme is produced means, right, the gene which produces this enzyme is active. This enzyme is active means what does it indicates? It indicates that desired gene is not inserted. Desired gene is not inserted means it becomes which cell, sir? It becomes a non-recombinant transformed cell. Therefore, non-recombinant transformed cell. What it becomes, sir? Non-recombinant transformed cell. It becomes what? Non-recombinant transformed cell. This is for which cell, sir? Non-recombinant. Non-recombinant transformed cell. Therefore, recombinant transformed cells are separated from non-recombinant transformed cells by insertional inactivation, blue-white selection. Here, what is inactivated? Beta galactosidase enzyme producing gene is inactivated when desired gene is inserted. So, insertional inactivation. When you are going to say that it is a recombinant transformed cell, if blue colored product is not produced. If blue colored product is not produced, it is a recombinant transformed cell. If blue colored product is produced, it is a non-recombinant transformed cell. Is this statement okay? Right? Recombinant transformed cell, right? Recombinant transformed cell. If it is a recombinant transformed cell, right, no blue colored product or no blue colored what's our colonies, right, sir? Non-recombinant what's our transformed cells. Non-recombinant transformed cells means you are going to get what's our blue colored product or blue colored colonies. Blue colored colonies. Is this statements okay? Right? Sir, to produce a what? To produce a blue colored product, to produce blue colored product, there should be right, number one, chromogenic substrate, right? Number two, second one, what's our beta galactosidase enzyme? Which enzyme, sir? Beta galactosidase enzyme. Listen here. Ampicillin used. What is used? Ampicillin used to select what? To select transformed cells. To select what? Transformed cells. To select transformed cells from non-transformed cells. From non-transformed cells. Is this statement okay? Right? Therefore, to select transformed cells from non-transformed cells, what was used, sir? Ampicillin was used, sir. Okay. Now, which cells are there, sir? Transformed cells. Right? Now, listen here. To select recombinant transformed cells. To select what, sir? Recombinant transformed cells from where? From non-recombinant transformed cells. From non-recombinant what? From non-recombinant transformed cells. Okay. To select what? Recombinant transformed cells from non-recombinant transformed cells, either tetracycline used, right, or insertional inactivation. Insertional inactivation. Therefore, by using ampicillin, first we separated transformed cells from non-transformed cells. Now we are having only transformed cells. But these transformed cells, they may be either recombinant transformed cells or non-recombinant transformed cells. How we are going to separate recombinant transformed cells from non-recombinant transformed cells are either by using tetracycline or for going insertional inactivation, that is a blue-white selection. Is this statements okay? Right? So we completed insertional inactivation. Right? Go for the last method. Right? What it is? Colony hybridization. What is this method, sir? Colony hybridization. Sir, in this colony hybridization method, probes are used. What are used, sir? Probes are used. Probes are used. Sir, what is the meaning of a probe, sir? Probe is a small fragment. Each and every word is important, therefore be careful with these words statements, right? Small fragment of what? Single stranded DNA or single stranded RNA to which a radioactive element is attached, to which a radioactive element is attached, and its sequence should be complementary. Its sequence should be complementary to at least a part of what's our desired gene. Complementary to at least a part of what's our desired gene. At least a part of a desired gene. See these statements. Sir, probe means it is a small fragment, sir. Small fragment of what? Either DNA or RNA. It should be what's sir? Single stranded. Okay, and to this small fragment of single stranded DNA or RNA, right, what we are attaching? A radioactive element is attached, and this probe sequence should be complementary to at least a part of what's our desired gene. At least a part of desired gene. Sir, probe is used to identify the desired gene. Probe is used to select the what's our desired gene. Therefore, probes used to select cells with which gene? Desired gene. Used to select, right, what's that? Cells with a desired gene. Is it okay? Right, we completed what's our colony hybridization. Is it okay? So we completed selection of transformed cells. Selection of transformed cells, right? Discussing steps of genetic engineering, right? In the steps of genetic engineering, go for the last step. What's obtaining foreign gene product. Obtaining foreign gene product. Okay. Sir, in first step, we isolated a desired gene, right? In first step, we isolated a desired gene. Isolated a what, sir? Desired gene. Isolated a desired gene. Sir, in second step, we inserted this desired gene into a vector. Insertion of desired gene. Where, sir? Into a vector. Which vector, sir? Cloning vector. We inserted the desired gene into a cloning vector. Then the cloning vector or vector with the desired gene is called as what, sir? rDNA. It means a recombinant DNA is formed. What is formed, sir? Recombinant DNA. What is another name of recombinant DNA? Chimeric DNA. rDNA means nothing but what, sir? Vector plus desired gene. Yes or no? Right? Vector plus desired gene is equals to rDNA. Now this rDNA is introduced into the what, sir? Host cell. Right? What, sir? rDNA. That is a recombinant DNA introduced. rDNA introduced into host cell. Now listen this statement. Sir, now where is the rDNA, sir? Inside the host cell. rDNA means what, sir? Vector plus desired gene. It means now where is the desired gene? Now desired gene is inside the host cell. Yes or no? Once rDNA is introduced into the host cell, the desired gene multiplies. Desired gene multiplies. It means gene cloning takes place. What takes place, sir? Gene cloning takes place. Is it okay? Therefore, once rDNA is introduced into the what, sir? Host cell. What happens? What happens, sir? Desired gene. Desired gene multiplied. Desired gene has multiplied. This multiplication of desired gene we are calling as what, sir? Gene cloning. We are calling this process as what, sir? Gene cloning. We are calling this process gene cloning. Now listen this statement. Sir, the ultimate aim of many many rDNA, that is recombinant DNA technologies, is to produce a protein. Which protein? Desired protein. Now see here how the desired protein is produced, sir. Now what is this one, sir? This is a desired gene. Now this desired gene is expressed. What is expressed? The desired gene is expressed. When desired gene is expressed, it produces a protein. When what is expressed? Desired gene is expressed, it produces a protein. Therefore, desired gene expressed. Desired gene expressed, right? To produce. Desired gene expressed to produce a protein. To produce a protein. Is this statement okay? Any doubts in these statements? Therefore, desired gene is expressed to produce what, sir? Desired gene is expressed to produce a protein. Is it okay? Ultimate aim of many what? rDNA technologies is to produce a desired what? Protein. But to produce a desired protein, the gene should express. Therefore, gene should express. Then only what is produced, sir? A desired protein is produced. Is this statement okay? But to express the desired gene, expression of what? Desired gene. Expression of desired gene inside the host involves many technical steps. involves many technical steps, Is this statement okay? Clear, right? Therefore, what we are saying, sir? Ultimate aim of many, ultimate aim of many rDNA, that is a recombinant DNA technologies, is to produce a desired protein, but to produce a desired protein, but to produce a desired protein, right? But to produce a desired protein, the desired gene should be expressed. What should be expressed? Desired gene should be expressed. Gene expression, it means expression of a gene. Expression. Gene expression involves many technical steps. Gene expression involves many technical steps. Now, sir, what is the meaning of gene expression, sir? What is the meaning of gene expression? Sir, what is the meaning of a gene? Generally, gene means it is a segment of what, sir? It is a segment of DNA. Gene means what, sir? Gene means it is a segment of DNA. Gene means it's a segment of DNA. Sir, first this gene produces what, sir? RNA. This gene is producing what, sir? RNA by a process called as transcription. What is that process called as, sir? Transcription. Gene produced what? RNA. What is this process called as? Transcription. Now this RNA produces a protein. RNA produces what, sir? Protein. This is called as a translation. What it is called as a translation. Sir, we want gene expression. Where is gene expression, sir? This itself called as gene expression. Therefore, gene expression means nothing but to produce a protein. Gene expression means to produce a protein, but gene cannot produce protein directly. So gene produced RNA by transcription. This RNA in turn produces protein by translation. Therefore, gene expression means to produce a protein. Gene cannot produce protein directly. So gene produces RNA by transcription. This RNA in turn produces protein by translation. Therefore, gene expression involves how many steps, sir? 2 steps. What and what, sir? Transcription and translation. What and what, sir? Transcription and translation. Therefore, gene expression. Gene expression means what? To produce a protein. To produce a protein. Is it okay? Gene expression involves how many steps, sir? Involves 2 steps. What are the 2 steps, sir? Step 1. What it is? Transcription. Step 2. What? Translation. Is this statements okay? Now listen here. Sir, to produce a desired protein, the gene should express. Sir, where the gene is expressing? It is expressing inside the host cell. It is expressing inside the host cell. Is it okay? Now listen this statement. When gene is expressed in a new host, when a gene is expressed in a new host, for example, I isolated insulin producing gene from my body. Insulin producing gene is isolated from human body. I inserted that insulin producing gene into a vector, cloning vector. rDNA is formed. Now I am introducing this rDNA into the host cell, for example bacterial cell. Now you tell me, desired gene belongs to human being, host is bacteria. It means host is new for the desired gene, yes or no? Actually the gene should express in the human body, but now where it is expressing? It is expressing inside the bacterial cell, yes or no? Because the gene what I isolated is insulin producing gene. Is it okay? Therefore insulin producing gene is isolated from human body. That insulin producing gene inserted in a cloning vector, rDNA is formed. That rDNA is introduced into the host bacterial cell. Now you tell me, where is the insulin producing gene? It is inside the host bacterial cell. It means where the insulin producing gene will express, it will express inside the host bacterial cell. But actually should be expressed in the human body. Should be expressed in human body, but now it is expressing in the host bacterial cell. It means it is expressing in a what? In a new host cell, heterologous host, new host. That's why, sir, when gene is expressed, what produced? Protein. That's why this protein produced is called as a recombinant protein. What is this protein called as? Recombinant protein. The desired gene, when expressed in a new host, the protein produced is called as a recombinant protein. The protein produced when desired gene is expressed in a new host. Desired gene is expressed in a new host, that is heterologous host. Then what is that protein produced called as? Recombinant protein. What is the protein called as a recombinant protein? Therefore, the protein produced when the desired gene is expressed in a new host, that protein is called as what, sir? Recombinant protein. Sir, to produce this recombinant protein in small quantities, right? To produce a recombinant protein in what, sir? Small quantities, we have to culture the host cells in lab. Is it okay? Right. Therefore, to produce what? Recombinant protein. To produce what? Recombinant protein in small quantities. Small quantities. What we have to do? Culture means grow. Culture host cells in lab. Therefore, to produce recombinant protein in small quantities, we have to culture the host cells in what's our lab. Whereas, to produce what? Recombinant protein in large scale, means industrial scale. To produce this recombinant protein in large scale, culturing should be done right in bioreactor. Culturing done in what's a bioreactor. Culturing is done in what, sir? Bioreactor. Culturing is done in bioreactor. Is this statement okay? Therefore, to produce recombinant protein in small quantities, culturing is done in the lab. To produce this recombinant protein in large quantities, culturing is done inside the bioreactor. Culturing is done inside the bioreactor. Is it okay? Right? Bioreactors. Right? Sir, commonly used bioreactor. Commonly used bioreactor. Commonly used bioreactor. What, sir? Stirred tank bioreactor. What, sir? Stirred tank bioreactor. Is it okay? Right? Sir, this what? Stirred tank bioreactor or stirring tank bioreactor. It may be simple stirred tank bioreactor or sparzed, right, tank bioreactor. Is it okay? Right? Sir, if you take a bioreactor, that bioreactor is either cylindrical or its base is somewhat curved. Is it okay? Right. Therefore, bioreactor. How the bioreactor is, sir? Bioreactor. Which bioreactor we are speaking about? Stirred tank bioreactor, right? Which bioreactor, sir? Stirred tank bioreactor. Sir, this stirred tank bioreactor, it is either what? Cylindrical or base curved. Sir, why we are taking it cylindrical or base curved, sir? Because what mixture, what content we are taking inside the bioreactor, that should be mixed. For that mixing, we are taking bioreactor either cylindrical or base curved, right? Which facilitates what? Which facilitates mixing. Which facilitates what, sir? Mixing of reactor content. It means what we are taking inside the bioreactor, for mixing of that content, right? Bioreactor should be cylindrical or its base should be, how, sir? Curved. Is this statement okay? Right? Now, sir, in this bioreactor, right, 100 to 1000 liters of culture can be processed. How much? 100 to 1000 liters of culture processed. How much culture is processed, sir? 100 to 1000 liters of culture is processed. Is this statement okay? Right? Now listen here. Sir, we are discussing what, sir? Bioreactor. Sir, bioreactors are vessels in which raw materials are biologically converted. Raw materials are biologically converted into specific products or individual enzymes, so on, using what? Using host cells. Using which cells, sir? Host cells. That host cells may be either plant, animal, or, right, bacterial cells. Even human cells also we can take. Is this statement okay? Right? Therefore, sir, bioreactors, Bioreactors are what, sir? They are containers, they are vessels in which what happens? In which raw materials, what? Raw materials are biologically converted to specific products. What are the specific products, sir? That specific products may be either proteins or individual enzymes using what, sir? Using a plant, animal, human or bacterial cells. Therefore, using either plant or animal or human or bacterial cells, biologically, it means using living organisms, right? Raw materials are converted into specific products in a vessel. That vessel is called bioreactor. Is it okay? Right. Sir, we are using living organisms, plant, animals, human, bacterial cells as host cells. It means biologically raw materials are converted into specific products such as proteins or individual enzymes in a vessel. That vessels are called as bioreactors. Therefore, bioreactors are vessels in which raw materials are biologically converted into specific products using plants, animals, human or bacterial cells. Sir, all this acts as what, sir? Host cells. But sir, whatever conditions required for culturing of these host cells, all the conditions should be artificially provided inside the bioreactor. Therefore, conditions required for what? For growth of culture. What are the conditions, sir? Such as temperature, pH, salt, substrate, vitamins and oxygen should be provided. All these conditions what are required for growth of the culture, all that conditions should be provided by the bioreactor. Sir, how bioreactor is providing these conditions that are required for growth of this culture, sir? How it is providing, right? All these conditions that are required for growth of the culture, right? Sir, here what are the conditions that are required for growth of the culture? Temperature, pH, salt, substrate, vitamins, oxygen, right? All this should be provided by the bioreactor. How, sir? Because bioreactor contains a temperature control system. It contains what, sir? A temperature control system. It also contains pH control system. pH control system. It also contains foam control system. And for delivering oxygen, it means to input the oxygen, to introduce the oxygen into the bioreactor, oxygen delivery system is also present. It even contains a sampling port. Sir, what is the use of sampling port, sir? Periodically, it means time to time, to remove small quantity of culture from bioreactor, this sampling port is used. Therefore, to remove what? To remove culture. To remove what? Small amount of culture. To remove small amount of culture from what? Bioreactor. From bioreactor time to time. Therefore, to remove small amount of culture from bioreactor time to time, what is provided, sir? Sampling port is provided. What is provided? Sampling port is provided. Right, sir. This bioreactor, what, sir? Bioreactor also contains a stirrer. Sir, what is the use of this stirrer, sir? Sir, stirrer, what it is doing means it is used for even mixing of what it is used for? It is used for even mixing of the content what we are taking inside the bioreactor, as well as oxygen availability. Oxygen availability throughout the bioreactor. Oxygen availability throughout the bioreactor. Therefore, sir, stirrer used for even mixing and oxygen availability throughout the bioreactor. Even mixing and oxygen availability throughout the bioreactor. What is provided? Stirrer is provided. What is provided? Stirrer is provided. Now listen here. Inside the bioreactor, what's there? Inside the bioreactor. Inside the bioreactor, culture is in continuous system. Inside the bioreactor, we can call it as a continuous culture system. Continuous culture system is maintained. What is maintained? Continuous culture system is maintained inside the bioreactor. Sir, how continuous culture system is maintained, sir? By removing the used up nutrient medium and by introducing the fresh nutrient medium into the bioreactor. Therefore, time to time, used up what? Used up medium removed and fresh medium introduced. Fresh medium introduced into what? Bioreactor. Into bioreactor. Therefore, used up is removed, fresh medium is introduced. So what happens means a continuous culture system is maintained inside the bioreactor, by which host cells are in log phase of growth. Are in which phase of growth, sir? Log phase of growth. Sir, what is the meaning of log phase, sir? L-O-G, logarithmic phase or exponential phase. This word is important. Therefore, sir, host cells are in which phase of growth, sir? Log phase, that is a logarithmic phase or exponential phase of growth. It means a rapid growth, fast growth of the host cells takes place. As the number of host cells increases, automatically the product what we are expecting from the host cells will be produced in large quantities. Is it okay? Right? This is concerned to what's a bioreactors. There is a stirring type of bioreactor. Okay? Right? Thank you.